Hello everyone, and welcome to today's presentation. I'm excited to have you here as we explore the fascinating topic of metabolism of xenobiotics. This is the first slide, serving as our cover, where we set the stage for what's to come. We'll be discussing how our bodies process foreign compounds, or xenobiotics, and the mechanisms involved. Over the next few slides, I'll guide you through the key concepts and the significance of this process. Let's get started. So, when we talk about xenobiotics, we're often referring to pollutants like dioxins and polychlorinated biphenyls, or PCBs, and their impact on living organisms. These substances are considered xenobiotics because they are foreign to the entire biological system. Essentially, they are artificial chemicals that didn't exist in nature before humans synthesized them. The word itself comes from Greek roots, xi epsilon u omicron sigma, meaning foreigner or stranger, and beta iota omicron sigma, meaning life. The suffix tau iota kappa omicron sigma indicates an adjective form. Xenobiotics are any chemical compounds that appear in a living organism, but are not naturally produced or normally consumed as part of that organism's diet. For example, in humans, most drugs fall into this category since we don't normally produce or consume them naturally. Understanding xenobiotics is important because they can have significant effects on health and the environment, especially when they accumulate or disrupt normal biological processes. Throughout this presentation, we'll explore how these foreign substances interact with biological systems and what that means for both ecological and human health. Let's talk about xenobiotics, which are substances that are either found in higher than normal concentrations or are entirely artificial, meaning they didn't exist before humans started synthesizing them. These substances can come from many sources, including drugs and environmental chemicals. Now, when our body encounters xenobiotics, it has a specialized process to handle them, called drug metabolism, which is divided into three main phases. In the first phase, phase I, enzymes like Cytochrome P450 oxidases come into play. Their job is to introduce reactive or polar groups into the xenobiotic molecules, making them more reactive or easier to modify later on. Next, in phase II, these modified compounds undergo conjugation, meaning they are linked to polar molecules. This step is catalyzed by transferase enzymes, such as glutathione S-transferase, which help attach these groups to make the compounds more water-soluble. Finally, in phase III, the conjugated xenobiotics may be further processed, but the key point here is that the body recognizes these modified compounds through specific transporters. These transporters then actively pump the xenobiotics out of the cells, effectively eliminating them from the body. Understanding this process is crucial because it explains how our bodies detoxify and clear foreign substances, linking directly to the overall theme of how we manage exposure to various chemicals. This slide doesn't contain enough information. That's why we're moving to the next slide. This slide doesn't contain enough information. That's why we're moving to the next slide. Drug metabolism is essentially how our bodies break down drugs and other foreign substances, which we also call xenobiotics. This process usually involves specialized enzymes that modify these compounds biochemically. One of the main goals of drug metabolism is to transform lipophilic, or fat-soluble, chemicals into more hydrophilic, or water-soluble, forms. This conversion makes it much easier for the body to excrete these substances through urine or bile. The rate at which this metabolism occurs is really important because it influences how long a drug stays active in the body and how strong its effects are. If a drug is metabolized quickly, its effects might not last very long. If it's metabolized slowly, the effects could be prolonged. And of course, this process isn't just about drugs. Our bodies also remove other foreign compounds, or xenobiotics, through what's called xenobiotic metabolism. So, understanding these mechanisms helps us grasp why drugs behave the way they do inside our bodies, which is key to effective pharmacology and personalized medicine. Now, let's talk about how the body handles xenobiotics, which are foreign substances like drugs or pollutants. This process mainly involves two steps: deactivation and excretion. Deactivation is where the body makes these substances less harmful, often through chemical modifications. Following that, excretion is how the body gets rid of these now less harmful compounds. Most of this activity occurs in the liver, which acts as the body's primary detox center. Once processed, the xenobiotics can leave the body through various routes, primarily urine and feces, but also through breath and sweat. These routes are essential for clearing out substances efficiently and keeping our internal environment safe. Understanding these mechanisms helps us appreciate how our body manages to protect us from potentially harmful external compounds. Hepatic enzymes play a crucial role in how our body handles xenobiotics, which are foreign compounds like drugs or toxins. These enzymes first activate the xenobiotics through processes like oxidation, reduction, hydrolysis, or hydration. Think of this as making the compound more reactive or easier to process. Once activated, the enzymes conjugate the metabolite with substances such as glucuronic acid, sulfuric acid, or glutathione. This conjugation step is important because it makes the xenobiotic more water-soluble, which facilitates its excretion. After conjugation, these compounds are typically excreted either in bile or urine, completing the detoxification process. An important group of enzymes involved here are the hepatic microsomal cytochrome P450 enzymes. These enzymes are especially significant for the pharmaceutical industry because they determine how quickly medications are broken down and cleared from the body. In essence, understanding these enzymes helps us predict drug metabolism and potential interactions, which is vital for safe and effective therapeutics. So overall, hepatic enzymes act as the body's detox system, transforming foreign substances into forms that can be safely eliminated. And they are especially important when developing and prescribing medications. Now let's look at how this process occurs in two distinct phases. We'll start with phase I, where the initial modifications take place, often involving reactions like oxidation. This phase prepares the compounds for the next step. Then, in phase II, we see the conjugation reactions, which are crucial for making these compounds more water-soluble and easier to eliminate from the body. During conjugation, key molecules such as glucuronic acid, amino acids like glycine, glutathione, sulfate, acetate, and methyl groups are involved. These act as conjugating agents, attaching to the target compounds to modify them. It's important to note that oxidation isn't the end of the story. It's followed by reduction and hydrolysis. These subsequent reactions help further process the compounds, ensuring they're in the right form for excretion. So, the compounds involved in conjugation play a vital role in detoxification, making sure harmful substances can be safely eliminated. This entire process highlights how the body efficiently manages and removes foreign or potentially harmful compounds, tying into our overall theme of metabolism and detoxification. In phase I of metabolism, a variety of enzymes work to introduce reactive and polar groups into their substrates. One of the most common modifications we see here is hydroxylation, which is catalyzed by the Cytochrome P450 dependent mixed function oxidase system. This enzyme system is particularly important because it allows the body to modify non-activated hydrocarbons by inserting an oxygen atom. This process can lead to the incorporation of hydroxyl groups or facilitate N, O, and S dealkylation of the substrate. The key point here is that these reactions are driven by the activity of P450 oxidases, which operate through a specific scheme involving the reduction of cytochrome-bound oxygen. This results in the formation of highly reactive oxyfuryl species, which are crucial for the oxidation process. The overall reaction involves NADPH donating electrons to reduce oxygen and activate it for substrate modification. It's also important to note that phase I reactions, often called no-synthetic reactions, include oxidation, reduction, hydrolysis, cyclization, and other transformations like decyclization or oxygen addition. These reactions primarily occur in the liver and are facilitated by mixed-function oxidases, especially the cytochrome P450 monooxygenase, or CYP. In summary, phase-I reactions are essential for making compounds more water-soluble and ready for further processing, all of which contribute to the body's ability to detoxify and eliminate various substances efficiently. Let's take a closer look at the key processes involved in phase-I reactions. First, we have oxidation, reduction, and hydrolysis. These are fundamental types of chemical reactions that modify substrate molecules. During these reactions, the molecules often undergo changes in their structure and polarity. An important point here is that the substrate's polarity increases, which makes it more water-soluble and easier to handle by the body. Additionally, these reactions help the substrate gain a suitable structure for the next phase, phase II, where conjugation reactions occur. Essentially, phase-I reactions prepare the substrate by introducing or uncovering functional groups, setting the stage for subsequent conjugation and detoxification processes. Understanding these initial modifications is crucial because they influence how effectively the body can eliminate potentially harmful substances. Let's take a closer look at some of the key enzymatic systems involved in oxidation processes. First, we have the cytochrome P450 dependent monooxygenases, or CYP enzymes. These are incredibly versatile and important for metabolizing a wide range of substances, including drugs and toxins. They function by incorporating one atom of oxygen into their substrates, aiding in detoxification and metabolism. Next, there's the flavin monooxygenase, or FMO. Similar to CYPs, FMOs also catalyze oxygenation reactions, but they tend to act on different types of compounds, often nitrogen and sulfur-containing molecules. Both CYPs and FMOs are part of the body's defense mechanism against potentially harmful substances. Then we have alcohol dehydrogenases, or ADH. These enzymes are crucial for alcohol metabolism, converting alcohols into aldehydes or ketones. This step is vital for processing ethanol and other alcohols efficiently. Moving on, oxidase monoamine enzymes are responsible for the oxidation of monoamines, such as neurotransmitters. This process helps regulate their levels in the nervous system, maintaining proper neural function. We also see the role of peroxidases in reducing co-oxidation reactions. These enzymes help mitigate oxidative stress by breaking down peroxides, preventing cellular damage. Finally, the reductase of NADPH in the cytochrome P450 system plays a pivotal role by providing the necessary electrons for the CYP enzymes to carry out oxidation reactions. NADPH acts as an electron donor, fueling these vital metabolic processes. Overall, these enzymes work together in a complex but coordinated way to facilitate oxidation, which is fundamental for detoxification, metabolism, and maintaining cellular health. Now let's look at prontosol, which is actually an inactive prodrug. This means that it needs to be metabolized into an active form to exert its therapeutic effect. In this case, the active drug is sulfanilamide. Most commonly, when we talk about reductive reactions in biochemistry, we're referring to the reduction of nitro groups and azo groups. These types of reactions are quite significant because they often occur in the gut flora or in the liver, which are key sites of drug metabolism. For example, prontosol itself undergoes reduction to release sulfanilamide, which is the active antibacterial agent. Other examples include nitrobenzene and similar compounds, which also undergo reduction to become active or sometimes toxic metabolites. So, in essence, reduction reactions play a crucial role in drug activation and metabolism, especially involving these nitro and azo groups. Understanding these processes helps us grasp how certain drugs are metabolized and how their activity can be modulated within the body. Let's take a closer look at hydrolysis, which is a fundamental concept in chemistry. Hydrolysis essentially involves breaking down a compound using water. In the context of our example, think about how aspirin is converted into salicylic acid through this process. When aspirin undergoes hydrolysis, water molecules interact with it, breaking the ester bond and resulting in salicylic acid and acetic acid. This reaction is important because it demonstrates how chemical substances can be transformed in biological systems or during manufacturing. Understanding hydrolysis helps us grasp how drugs are metabolized or how materials degrade over time. So, in summary, hydrolysis is a crucial reaction that involves water breaking chemical bonds, leading to the formation of new compounds, which plays a significant role in both natural processes and industrial applications. Let's take a closer look at the CYP enzyme family in humans. This family is actually the most important enzymatic system involved in phase I drug metabolism. When we talk about phase I, we're referring to those initial modifications that prepare drugs and other compounds for further processing. Research from literature databases shows that CYP enzymes are responsible for the vast majority of these reactions, around 90 to 95%. That highlights just how central they are in metabolizing xenobiotics, which are foreign substances like drugs or environmental pollutants. While CYPs are the main players, it's worth noting that other enzyme systems can also participate in hepatic phase I metabolism, but their role is generally less prominent. Cytochrome P450s, which are part of this enzyme superfamily, are especially crucial. They handle a wide variety of reactions: oxidative, peroxidative, and reductive, that process both endogenous or internally produced compounds like steroids, bile acids, fatty acids, prostaglandins, and leukotrienes, as well as xenobiotics, including most therapeutic drugs and pollutants from the environment. So, understanding the cytochrome P450 system is key, because it underpins much of how our bodies process and detoxify a broad spectrum of chemical substances. Let's take a closer look at the molecular structure of cytochrome P450. This enzyme is fascinating because of its complex and highly specialized architecture. At its core, it contains a heme group, which is essential for its function in metabolizing various substances. The heme contains an iron atom that plays a crucial role in the oxidation process. Surrounding this core, the protein structure creates a specific environment that allows the enzyme to interact with a wide range of substrates. The overall shape and flexibility of the molecule are key to its ability to catalyze reactions efficiently. Understanding this structure helps us appreciate how cytochrome P450 can handle such a diverse array of compounds, which ties directly into its importance in drug metabolism and detoxification processes. So the structure isn't just a static picture. It's a dynamic framework that underpins the enzyme's versatility and effectiveness in the body. Let's walk through the initial step of the P450 catalytic cycle. It all begins when the substrate binds to the enzyme's active site. This binding displaces the water molecule that was attached to the heme's distal face, which is the sixth ligand. As a result, there's a shift in the iron's spin state from low spin to high spin, and this is a key change because it affects the enzyme's electronic properties. This spin shift also causes a change in the redox potential of the heme, making it more receptive to accepting an electron. Consequently, a redox partner can transfer its electron, reducing the ferric iron to ferrous or Fe-Roman-2. This reduction is crucial because it primes the enzyme for oxygen binding. Once oxygen binds to the ferrous heme, it forms a superoxide species, often called Fe-superoxo. This species then receives a second electron, which further reduces it, followed by protonation. This process leads to the formation of a ferric hydroperoxide, known as compound 0. Protonation causes this intermediate to collapse, releasing a molecule of water and generating compound I. This is the critical oxidizing species that actually performs the substrate oxidation. It does so either through a radical rebound mechanism or a concerted process, ultimately inserting an oxygen atom into the substrate. Throughout this cycle, the enzyme cleverly harnesses changes in spin states, redox potentials, and protonation steps to facilitate precise and efficient oxidation reactions. Truly an elegant example of nature's catalytic mastery. Now, let's take a closer look at the P450 catalytic system. This system is really fascinating because it plays a crucial role in the metabolism of a wide variety of substances, including drugs, toxins, and even some endogenous compounds. The key point here is that P450 enzymes are heme-containing monooxygenases, meaning they incorporate one atom of oxygen into their substrates while reducing the other oxygen atom to water. What's particularly interesting is how versatile and efficient this system is. It involves a series of steps where electrons are transferred from NADPH through a series of redox partners, ultimately reaching the P450 enzyme. This electron transfer is vital because it powers the catalytic cycle, enabling the enzyme to perform oxidation reactions. The overall process is highly coordinated, ensuring that substrates are modified in specific ways, which is essential for detoxification and metabolism. Connecting this to the broader theme of our presentation, understanding the P450 system helps us appreciate how organisms manage chemical diversity and adapt to their environment through incredible enzymatic machinery. Now, let's take a closer look at the phase II conjugation reaction. This is a crucial step in drug metabolism because it helps make compounds more water-soluble, which in turn facilitates their excretion from the body. During this phase, the body attaches specific conjugate groups to the drug or its metabolites, such as glucuronic acid, sulfate, or glutathione. These conjugation reactions significantly increase the polarity of the compound, reducing its ability to cross cell membranes and making it easier for the kidneys to eliminate. It's important to note that phase II reactions usually follow phase I modifications, which often introduce reactive groups. Overall, conjugation reactions are vital for detoxification and clearance of many substances, helping to maintain homeostasis and prevent potential toxicity. This step underscores the body's remarkable ability to process and eliminate foreign compounds efficiently. Now, let's talk about drugs like antibiotics. These are considered xenobiotics in humans because our bodies do not produce them naturally, nor are they part of a normal diet. When these substances enter our system, our body needs to handle them properly, and that's where xenobiotic metabolism comes into play. Xenobiotic metabolism is essentially a set of metabolic pathways designed to modify the chemical structure of these foreign compounds. The main goal here is to deactivate them and eventually eliminate them from the body. Most of this process occurs in the liver, which acts as the primary detox center. The routes through which these xenobiotics are excreted include urine, feces, breath, and sweat. The process is largely driven by hepatic enzymes, which first activate the xenobiotics through processes like oxidation, reduction, hydrolysis, or hydration. These modifications make the compounds more water-soluble and easier for the body to eliminate. Understanding this process is crucial because it highlights how our bodies maintain chemical balance and protect us from potentially harmful substances, especially when we take medications or encounter environmental chemicals. This metabolic pathway is a key component of our overall health management and drug safety. So, after a secondary metabolite has been formed, it can be conjugated with molecules like glucuronic acid, sulfuric acid, or glutathione. This conjugation process actually makes the compound more water-soluble, which helps facilitate its excretion. The metabolites are then eliminated from the body through either bile or urine, completing the detoxification process. Now, shifting gears a bit, let's talk about environmental xenobiotics. These are foreign substances in our environment that have biological activity. They're basically pollutants with structures that can interact with biological systems. Some of the major classes include polycyclic aromatic hydrocarbons, halogenated aliphatic and aromatic hydrocarbons, nitroaromatic compounds, azo compounds, estriazines, organic sulfonic acids, and synthetic polymers. These are important because they often pose risks to health and ecosystems due to their persistence and toxicity. And this is where microbial degradation comes into play. The focus of this part of the discussion is on how microbes break down these complex pollutants. Understanding these pathways and mechanisms is crucial, as microbial activity can help detoxify and remove these harmful compounds from the environment, aligning with our broader goal of understanding biological systems and their role in pollution management. En esta última slide, quiero destacar algunas referencias clave que respaldan la información que hemos revisado. Primero, tenemos un artículo de Esteves, Roof y Cranendonk de 2021. Ellos explican muy bien el papel central del Cytochrome P450 en el metabolismo de xenobioticos. Y su revisión es fundamental para entender la importancia de esta familia de enzimas en la biotransformación de sustancias extrañas al organismo. Luego, está el trabajo de Hartman y Colgas de 2021, que se enfoca en cómo Conorrhabditis elegans, un nematodo muy utilizado en estudios toxicológicos, metaboliza y transporta xenobioticos. Esto nos ayuda a entender cómo diferentes organismos manjan estas sustancias, y que podemos aprender de estos modelos. También aparece una referencia de XP de 2018, que proporciona instrucciones para el uso de ciertos instrumentos o metodologías, aunque en nuestro contexto, refuerza la importancia de seguir procedimientos estandarizados en el estudio de estos procesos. En conjunto, estas referencias subrayan la relevancia del metabolismo de xenobioticos y la función de enzimas como el Cytochrome P450 en diferentes organismos. Nos ayudan a comprender mejor cómo estas enzimas influyen en la detoxificación y en la farmacocinética, aspectos clave en la investigación y en la aplicación clínica. Let's look at some recent research that highlights the importance of glutathione transferases and related enzymes. First, Perperopoulou and colleagues in 2018 reviewed advances in protein engineering and biotechnological applications of glutathione transferases. They emphasized how these enzymes are crucial in detoxification processes and have potential uses in biotechnology. Moving on, Islam et al., also in 2018, conducted a comprehensive genome-wide analysis of the glutathione S-transferase gene family in potatoes. They examined how these genes are expressed in different tissues and under various conditions, which helps us understand their roles in plant stress responses and overall health. Gong and team in 2017 explored genetic polymorphisms in cytochrome P450 oxidortictase, another key enzyme involved in drug metabolism. They showed how variations in this gene can influence drug efficacy and safety, which is vital for personalized medicine approaches. Finally, Nisar et al. In 2017 focused on glutathione S-transferases in the context of colorectal cancer. They discussed how these enzymes' biochemistry and genetic variability might affect cancer development and progression, highlighting their potential as biomarkers or therapeutic targets. Altogether, these studies underscore the interconnected roles of these enzymes in health, disease, and biotechnology, reinforcing the overarching theme of our presentation. Understanding enzyme functions and their applications can lead to significant advances in medicine and agriculture.